APOE4 gene found in chromosome 1. It affects 1 in 8,000 to 10,000 people, being most prevalent in children, and it is due to the accumulation of fatty substances produced during the vision cycle in the macula that is a part of the retina essential for vision. Stargardt disease falls under the category of macular dystrophy and is also known as an inherited retinal disease. And some of the symptoms of this condition are photophobia or light sensitivity, as well as loss of color perception and vision loss. So how and why does this disease happen? Before addressing this question, I would like to go into the introduction to genetics. So we know that DNA is used to synthesize proteins. DNA is then further organized into different genes that code for different proteins, such as enzymes like amylase in our saliva, or hormones like insulin in our blood to lower our glucose levels. Proteins are synthesized from DNA in two different steps, known as transcription and then translation. So DNA is composed of nucleotides. Nucleotides are the basic building blocks of nucleic acids. As we can see here, they are composed of a phosphate group, as well as a ribose sugar, and then a nitrogenous base. It is that nitrogenous base that determines whether our nucleotide is going to be a cytosine, a guanine, an adenine, or a thymine. I would also like to note that in RNA, which is different from DNA, uracil replaces thymine. As we can see here, with the base pairing of DNA, cytosine always binds with guanine, and adenine always binds with thymine, or uracil in the case of RNA. So DNA is composed of exons and introns. Exons are the sequences that are used to synthesize proteins, while introns are the sequences not used to code for proteins. It is in the process of transcription that an initial copy of the gene is made, containing both introns and exons. And it is through the process of splicing that the introns are removed and the exons are put together into what we call our final mature mRNA that is ready for translation and protein synthesis. So I want to show you here an example. Imagine this was our gene sequence. We would have on top our template strand and then underneath it our coding strand. Out of the template strand, we would get our copy of mRNA, which is composed of the exons, in the middle the intron, and then another exon. It is through the process of splicing that the spliceosome complex comes in. It is the protein in charge of reading the splicing sites between the exons and the introns, and then splicing it to remove the introns and put the exons together into our final mature mRNA. So, as of recently, introns were often referred to as part of junk DNA, and scientists thought they had no purpose. But we know now that that isn't entirely true. And while we're still not 100% certain of all their functions, we do know that they have many effects on different DNA processes, like splicing. Even more, variations in these DNA sequences can lead to alterations in this process, as well as defects in the protein being synthesized. An example of these variations are deep intronic variants. I would like to note that while there are many other types of variants in intronic or non-intronic that also lead to disease, we're only focusing on these types of variants for this research. So deep intronic variants are mutations in intronic sequences of DNA that lead to a disease or a pathogenic phenotype of that gene. They can cause a defect in the protein being synthesized due to the inclusion of a pseudoexon in the mature mRNA. But what is a pseudoexon? A pseudoexon is an intron sequence that is included into the mature mRNA and is interpreted as part of an exon. I want to show you an example here. In yellow, we would have our exons, and in blue, we have our intron. This would be the pre-mRNA copy. It is in splicing, as we can see here, that a part of that intron sequence was included into our mature mRNA, and that underlined would be the pseudoexon. This leads to a different protein being synthesized, which can be pathogenic and can lead to disease, in this case, Stargardt disease. That is where this research comes in. There is a huge focus nowadays on identifying all the types of variants and their effects. An example of this is the novel C.161-395 G2A deep intronic variant in the ABCA4 gene that it was recently discovered in 2023 by Karate et al. So we know that the ABCA4 gene is related to the production of ATP binding cassette transporter. It is a protein in charge of removing harmful substances produced during the vision cycle away from the macula. We know that this variant that was just discovered includes a 91 nucleotide long pseudoexon in our final copy of mRNA that we can see in blue. So this research is going to focus, because, no previous research has been done on this variant and its effects. And this research is going to fill that gap in the literature. So now that we know what problem this variant causes, we need to think, how could we solve it? So what about using antisense oligonucleotides? Oligo means multiple and antisense means complementary. So antisense oligonucleotides means a bunch of complementary molecules, of complementary nucleotides. So I want to show you an example here. Imagine this was our sequence of our nucleotide that was complementary to our target mRNA within the pseudoexon. The AON would bind to the sequence because it is complementary to it. So when we reach the process of splicing and our spliceosome comes in, it wouldn't identify the splicing site included by the pseudoexon, and so it is not included into the mature mRNA, and hopefully the splicing defect is corrected. So these molecules were first developed by Saminek and Stepherson to inhibit the replication of mRNA of Ross-Sarcoma virus, and so far 10 AON therapies have been approved by the FDA. AONs have been found to be particularly good at targeting intronic variants that lead to this inherited retinal disease, Stargardt disease. So then all this information led me to my research question. How can the splicing defect caused by the C.161-395 G2A variant in the ABCA4 gene be corrected by designing antisense oligonucleotides? So when designing the AONs, we need to take into account certain unchanging parameters. These are the length, the number of nucleotides that composes the AON, the GC content, how much guanine and cytosine the AON has, the melting temperature, it's the temperature at which the molecule degrades in our body. Then the free energy just looks at the stability and capability of reaction of that AON. The free energy of the AON-AON complex looks at whether these molecules will bind to themselves. And then the binding energy determines whether our AON is fit to target our target sequence. It is through a literature review that I found that 3 of these parameters have the most impact on the effectiveness of these molecules. It is the GC content, the melting temperature, and the binding energy. Since they are the most significant when determining the effectiveness of the AON, I'll use them to filter down my initial big number of molecules. So all the steps for my methodology were retrieved from the chapter by Garanto and Collin from the book Retinal Gene Therapy. I decided to use this methodology since it was cited very often among different scientific papers of the field, and I saw little variance amongst the parameters for different scientists that developed AONs for their own research papers. It is here that the best target regions for these molecules were determined. These are the open-closed regions within the pseudoexon and the SC35 motifs that I will explain further in the presentation. And for all of this, designing the AONs and finding these best target regions, I used different software like OligoCalc, RNA structure, that I will also refer to further in the presentation. So for my methodology, my first step was identifying the pseudoexon included by this novel variant using the information provided by the article source. Then when I had this, I analyzed the region of interest to find my optimal target regions. when I had this, I designed a significant number of ions that targeted these SC35 motifs and the open-closed structures. And then I used different theoretical ranges to narrow down the number and in order to find the most effective ones. And finally, I ended up with 6 final ions that I will be proposing as optimal for further research. So my first point was identifying my region of interest and the pseudoexon. With the help of my mentor, I was able to retrieve the sequence of the ABCA4 gene, as well as the specific location of the pseudoexon. As we can see, it is the sequence in blue. And then, as specified in my methodology, I decided to add the 50 nucleotides flanking it and used the 191 nucleotide long sequence as the region of interest that my ions will be targeting. So for the optimal target regions, the open-closed structures, as we can see, this plot just shows which parts of the sequence are open or binded to each other that I will use then for designing the ions, and the SE35 motifs using the ESE finder. I only looked at the SE35 values and most specifically the ones included within the pseudoexon and the top 2 ones since they had the highest score, which means should be the most optimal. So then on to the design of the antisense oligonucleotides. Here I have my sequence of the sequence of my region of interest, and then I'm going to use as an example target region 2. As we can see, it is targeting an optimal region since it is underlined. And to get the complementary ion, we need to remember that DNA is read from 3' to 5' and is written from 5' to 3', which means that to determine my ion, I went that way and did the complementary sequence, which gave me ion number 2. So then, once I had the sequences for my ions, I put them through oligocalc to analyze their length, their GC content, and the melting temperature. As you can see, we have determined ranges that our ions need to fall in. Ion number 2 falls within all these ranges. So far, this ion would be a good ion, an optimal ion. It's looking good. Then for the free energy of the ion, I used AllSub, and this software calculates the lowest free energy structure for this molecule. So I put my sequence, and it gave me what the ion would look like, and then my value for the free energy, which also is higher than the determined range, -5. So far, it's still good. Then for the free energy of the ion-ion complex, I used the same software but with a different option, bifold, which just compares the sequences of the ion to determine whether or not they will bind to themselves. And it gave me also a value that is higher than the appropriate range. So still good with ion number 2. Then for the binding energy, first of all, I needed to determine the free energy of the region of interest. Still using the same software, and it gave me this huge structure, as we can see here, and then the free energy of the whole region. Then when I had this value, I did the same. Instead, I used bifold to compare the 2 sequences of my ion and the region. And it also gave me the free energy for this ion region complex. When I had these 2 values, then I went on and used the formula given by my methodology to calculate my final binding energy. As we can see, this is our formula. We just have to subtract the energy of the region minus the energy of the region plus the ion to get our binding energy and determine whether or not the ion is fit for this region. As we can see, it falls under the parameter between 21 and 28. So far, this ion should be optimal for this region. And I did this over and over 50 times for each of the ions that I designed for this pseudoexon. As we can see, this was the spreadsheet, and as I mentioned before, I used all of these parameters to filter down the number, which led me to my final 6 ions. As we can see, it is ion number 2, 4, 27, 28, 31, and 48. That is the target sequence that is the target sequence, and then the ion sequence, which scientists, when seeing these conclusions, should be able to use to further analyze the ion sequence and determine whether or not it is actually effective. So, although these ions are theoretically effective, a first screening in vitro and in vivo testing should say whether or not these ions are actually effective when targeting these pseudoexon. But so far, for this research that just looks at whether these ions are appropriate or not, on basing it on ranges, the proposed ions should be theoretically optimal to correct the splicing effect of the C.161-395 T2A variant. Thank you. So I think the most important skill that I learned is not getting stuck when I make mistakes. Because as you can see, this is a fairly complex topic. And when I got to working with it, I didn't know much more than you do. I had just grade 12 biology, which is pretty basic knowledge about genetics. So there were a few times that I couldn't understand certain things, certain concepts. And when I got into the actual design of the ions, many times I got stuck, and well, I needed Alex's help to try to tell me what I was doing wrong and how to fix it. So I think the most important part was that I didn't get stuck on the mistakes that I did, and I was quick trying to solve them, because it can get very frustrating when you don't know much about what you're talking about, and you're trying to catch up in a really short period of time. And you need to know how to get through those feelings of frustration and then finally get the work done as best as you can, and just try to not overthink it, I guess. So the problem is not making mistakes, just how you solve them. I think that's what this research taught me.
Especially when your research teacher has very little understanding of what you're doing. Erin, could you please go ahead?
Great. Well, I just want to say first that was such an amazing talk. You've clearly done a lot of work. You know, you're saying you just have your level of understanding, but it far exceeds mine at this point. You've really done an amazing job. And so the question that I want to ask is more, I know the research team knows it, so Alex knows, you know the process, but I think it's important for you to kind of explain to your peers that research evolves and changes, right? So my question is, how did your initial exploration of the scholarly conversation lead to your final research project?
Well, so first of all, I guess when I looked into this particular field, I found that scientists followed different steps. First of all, it was identifying the problem, or in this case, the generic variant that was leading to disease. And then it was trying to find a solution for it. There are many different types of genetic therapies, but well, Alex is an expert on ions, so I started looking at how antisense oligonucleotides work. And once I got my gap for this research, right, this is the novel variant, I looked more into what was different ion therapies and how they worked and how they addressed all similar types of variants. And then that's when I knew that maybe I could design these ions to target the problem that I am addressing in the research.
Okay. Can you ask a question?
Absolutely, yeah. So there's no such thing as a perfect method, ever, right? So my question for you is, how did the limitations of the method that you used, and you mentioned that it was cited by a bunch of different papers as well, which is important, but how did the limitations of the method influence of the method or of the data that you accumulated, influence your understanding of your findings?
Well, I mean, yes, you said that this methodology was cited many times across different fields, across different scientific journals. And well, I knew that because I didn't have access to a lab, all my conclusions were going to be drawn from theoretical ranges and theoretical conclusions, right? So this methodology seemed the most appropriate since it focuses on the design of the ions and not the first screening on whether or not they will be effective when testing them in vitro. So I guess one of the limitations of this research is the fact that all my conclusions are theoretical, because we know that it's not all whether or not these ions fall under the appropriate range for length or GC content or melting temperature. You may test them and find out that they do nothing to correct the splicing defect. So I guess the limitation is that, well, I can't actually know whether or not they are appropriate. And although they do fall under the appropriate ranges and seem optimal, you may get to the lab and find out that these ions are not useful at all, and it was a waste of time. But I guess, yeah, that is the main limitation. And it is true that when going through different articles about the topic, all scientists first design the ions and then go to the first screening, which I am not able to do. So I guess not knowing whether these ions will work or not is one of the limitations of the research.
Thank you. Great job.
Thank you.
Okay. Absolutely. So with that, we can turn it over and have like open questions, real questions.
Okay.
So I'm going to give first crack at that to Alex and Aaron. Do you have any questions that you would like to ask?
Maybe can start. Oh, okay, sure. So many of the techniques that you used are widely applicable to oligonucleotide or kind of DNA and RNA research. So just for context, Alex and I both work with DNA, both in a non-traditional kind of not genetics way, but we're building tools, therapeutics tools. And so I'm wondering, when you were doing your research and kind of using these tools and learning about kind of all the different background that was required, if you just found any other fields interesting, if there's anything that you want to pursue, or if you want to keep pursuing kind of ASOs or AONs, what did you find there?
Honestly, everything about genetics is interesting to me. I mean, it's amazing how from DNA we are what we are. I mean, it blows my mind, honestly. And it is true that ions are very, very interesting. I had a lot of fun doing this research, but if I had to choose maybe different topics, I like how splicing works. It's a very interesting process, especially alternate splicing, how we can make different proteins from the same gene. I found that particularly interesting because I had no idea that worked like that. And I guess, I mean, honestly, everything interests me. Genetics is particularly my curiosity, but everything about the workings of the human body, not only DNA related stuff, I think it's amazing. So, yeah.
All of the above.
That's great.
Yeah.
That's awesome.
And from all the tools and programs you have to use, you already mentioned before, limitations and frustrations, even you mentioned that word. So what was the most challenging aspect that you, besides the fact that you were not familiar with the topic, but I mean, when you were learning these particular softwares, that some of them are the explanation of what the score means is usually written in a way that even the experts do not really get what it is, but how did you deal with that, and what did you find extremely difficult that you said, well, at this moment I want to give up?
Well, I mean, at the beginning, when I had zero knowledge and I found out what I was going to do, it was kind of, I kind of freaked out because I mean, I went through the chapter for my methodology and all the softwares that I had to use, all the parameters. I had no idea what the parameters meant. So that was kind of a huge, block for me because I had to catch up really quick. But besides that, I think the hardest part for the designing of the ions was how to do the complementary sequence for the ions. You know that because I first of all did 20 or so ions, and then I found out that they were completely wrong because I read the sequence incorrectly. I didn't know that coding strand means that it is from 5' to 3'. So I did the complementary of things that I didn't have to do, and I had to start over again, and I needed your help to visualize it, because that's the thing. DNA, well, you've seen, it's all A, C, G, A, U, and to me, that's very confusing, especially because it's very hard visualizing what I'm actually doing. So I guess retrieving the sequence and finally designing the AIONs appropriately, that was one of the main challenges.
We're just going to excuse our biology class here because the bell went and they have their next class to get to. But I have a question for you.
Yeah. Thank
Just give
you.
him 1 second. By the way, great job.
Thanks.
The hard part's over.
Yeah.
It was really good.
Thanks. I went a bit too fast, but.
That was powerful. That was powerful.
I'm pretty sure Kenny did that way.
Yeah, it was pretty really well made. Oh. Yeah, understood. Yep, just leave that behind and we're good. And you can shoot me an email if you have any final thoughts. Just quickly, is the therapy that you're suggesting, or like this kind of therapy, is that a like permanent solution to the splicing defect for this disease, or is this something where like will eventually be replicated and basically outcompete or overwhelm the existing defective DNA RNA, or is this something where you have to go back for like ongoing sort of therapy?
Well, I mean, I haven't looked as much at how the dosing of the therapy goes, but I did understand that these AIONs, they won't fix the problem completely. Well, I mean, if you already have a Stargardt disease and you've already have the symptoms and your macula is already degenerated, I guess the AIONs will alleviate the symptoms, but it will not complete or reverse the disease. And from what I understood, the AIONs are not like a one-time solution like gene editing where you change the DNA and that should be it. I do believe, I'd say, I haven't done much research on this part, but I'd say, yeah, you need to like go back for, I don't know, doses.
And I realize
periods
that's
of time.
not the scope of your research, so I'm not expecting you to be an expert on this. I'm just asking out of my own curiosity.
Yeah.
Okay.
Okay.
All right. Are we all good? Any final questions from anyone online?
I don't have a question, Irene, but I just wanted to clarify when we were talking about working with the complementary DNA being tricky, and Alex and I were laughing. It's because anyone who's worked with nucleic acids has had a silly story about like looking at the wrong direction, looking at the wrong complementary, reading it wrong. So we're right there with you. You're officially a nucleic acids chemist.
Yay.
Pretty soon we'll have AI to do all that for us, right?
Yeah, thankfully. It'll save us the frustration, right, and the trouble.
Okay. Unless you have any final thoughts or comments, feel free to shoot me an email with that feedback form. I'd greatly appreciate that. And otherwise, I wish you were here in person to thank you, but thank you so much for giving Irene this amazing opportunity and for all the support that you gave her.
Yes, thank you.
We really appreciate it.
It was a pleasure, and again, I want to congratulate Irene because what she did is really complicated for the high school student. I mean, even master students, when they come to the lab, they struggle with this kind of research. So she did brilliantly.
Okay.
Thank you.
Very much.
So you should be proud.
Thank you.
I agree.
I really thank you for your help. It was an amazing project. I'm really happy. So thank you both.
All right, well, with that, I'll say take care. Good night, Alex, and good afternoon, Aaron. All right, take care.
Thank you. Bye.
Bye.
Bye, take care, guys.
Bye.
There you go.
Okay.
You did it.
Thanks.